Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
If you, like us, spend time thinking about health and resilience, especially as we navigate a world where it feels like multiple respiratory illnesses are always making the rounds.
Colds, the flu, you name it.
Exactly. You're constantly looking for that edge, that reliable defense for your immune system.
No.
Well, what if I told you the answer wasn't waiting in some hyper-optimized lab, but maybe in a jar of traditional fermented Korean cabbage?
We are, of course, talking about kimchi. For centuries, it's been celebrated as a healthful food, a cornerstone of Korean culture and diet. But a truly groundbreaking study it's fundamentally shifted our perception.
How so?
It's moved kimchi from the category of just a general healthy food to something much more specific, a certified precision regulator for the human immune system.
And that is the mission of this deep dive. We're going to unpack the findings from research done by the World Institute of Kimchi, or WICHEM. This culminated in a publication in the journal NPJ Science of Food, and they didn't just do a standard dietary trial.
No, not at all. They combined a 12-week clinical study with really the most advanced analytical tool we have available today: single-cell genetic sequencing.
And that level of scientific resolution is why this study is so important, globally significant, really.
It is. Before this work, trials that looked at fermented foods could basically tell us two things: either how they changed the gut flora or
how they affected broad metabolic markers like cholesterol or weight.
Exactly. The connection to the immune system was always sort of inferred. It was indirect. This research changes that entire picture. It is the first study in the world to map the immunological effects of kimchi directly at the single-cell level.
The initial surprise here, the core takeaway that you really need to internalize before we dive into the complex mechanics, is that kimchi isn't an immune booster in that simple sense.
It doesn't just indiscriminately step on the accelerator.
Right. It acts as a sophisticated dietary immunomodulator. It simultaneously enhances crucial defense functions while actively suppressing the kinds of excessive, unnecessary immune responses that lead to chronic inflammation.
Think of it less like an emergency siren and more like a perfectly tuned, high-resolution surveillance system.
That analogy of a sophisticated thermostat is really key here.
It is, because in clinical immunology, indiscriminate boosting, just shoving the entire system into high gear, that can be dangerous.
You risk autoimmunity.
And you can exacerbate inflammatory diseases. What kimchi does, as this data reveals, is it calibrates the system. It makes sure the defense mechanisms are strong and responsive, but the regulatory brakes are also optimized.
So let's unpack how the researchers were even able to see this level of precision, because the method itself is pretty revolutionary for nutritional science.
Okay, so to isolate the effects of the kimchi, they used the gold standard of clinical research, a 12-week randomized, double-blind, placebo-controlled study. They enrolled 90 otherwise healthy adults.
And the choice of who they studied is important here. They focused on adults who were overweight, with a body mass index, a BMI, between 23 and 30. Why pick that specific group for an immune study?
That's a great question. It's because in individuals with higher BMIs, there's often a baseline state of low-grade chronic metabolic inflammation.
So there's already some static in the system.
Precisely. This is a population where the interaction between diet, metabolism, and immune signaling is often heightened and, a little bit dysfunctional. So if a dietary intervention can correct that low-level inflammation while also improving defense, it proves a powerful regulatory effect, not just a simple stimulation.
So the intervention had a measurable problem to solve from the get-go. These participants were then split into 3 groups.
Correct. And for the most expensive high-resolution analysis, the single-cell sequencing, they selected 13 participants from each of those groups.
And what did they consume?
For the full 12 weeks, they consumed a daily dose of a standardized powder supplement.
A powder? Why not just kimchi from a jar?
Using a powder ensures the dosage is exact and consistent. It removes all the variability you might get from homemade or even store-bought kimchi, where fermentation levels can fluctuate day to day.
Okay, that makes sense. So group 1.
Group 1 was the placebo. They got a capsule that matched in weight and texture but didn't have the active compounds.
And group 2.
They received what's called spontaneously fermented kimchi powder, or SK. This is meant to represent traditional natural fermentation driven by whatever microbes are already on the cabbage.
The old school method.
Exactly. And the third group received starter culture fermented kimchi powder, or LMSK. This version was inoculated with a specific optimized strain of lactic acid bacteria.
So this design allowed them to test if an engineered or, let's say, optimized fermentation provided better outcomes than the traditional method.
That's right.
Now we get to the powerhouse of this research, single-cell transcriptomics analysis. Let's just call it single-cell sequencing, but we should really take a moment to understand why this is such a game changer.
Okay. So traditionally, when scientists analyze the immune system, say, measuring cytokine levels, they do what's called bulk testing.
Bulk testing.
They take a sample of your blood's immune cells, all the key players, T cells, B cells, monocytes, and then they essentially grind them all up together and measure the average activity of that whole population.
It's like taking the average temperature of a stadium full of people.
Mm-hmm.
You might know if the crowd is generally running hot or cold,
But you have no idea if the fever is being caused by a small group of 10 people in one section who are panicking, while everyone else is perfectly calm.
Right.
Single-cell sequencing completely changes that. By isolating and analyzing each individual cell, thousands of them from each blood sample, they could track the distinct genetic instructions being activated or deactivated inside one T cell or one monocyte completely separately.
They could see who was talking, how loud they were talking, and what they were saying.
At a level of detail that was, just impossible in human nutritional trials before this.
This is a huge investment for a nutrition study. It says a lot about their commitment. Why is applying this specific tech to nutrition so revolutionary?
Because nutrition is inherently complex. When you eat kimchi, you're not just eating one molecule, you're introducing thousands of compounds, live microbes, their metabolites, fiber, vitamins.
Into an already complex system.
Right. Older bulk methods would have just detected a general inflammation marker going up or down. We wouldn't know if that was good or bad. Single-cell analysis let them see the precision. They could see that one type of cell was turning on its defense genes, while a different type of cell was simultaneously turning on its regulatory or calming genes.
So the high-res analysis wasn't a luxury. It was essential to prove kimchi was a regulator, not just a booster.
Precisely. They collected blood before the trial and again after 12 weeks. And comparing those 2 points in time let them see the subtle but profound shifts in each person's immune landscape, confirming that core finding.
Okay, so with that high-resolution data, let's zero in on who in the immune system was most dramatically impacted by the kimchi. First up, the antigen-presenting cells, or APCs.
These are the immune system's intelligence gatherers. They're the command and control center.
Like the Border Patrol agents.
That's a great analogy. Their primary function is surveillance. They recognize external invaders, a piece of a bacterium, a viral protein. They engulf it, break it down, and then they display fragments of that invader on their surface to signal the alarm to the rest of the immune system.
And the study showed the function of these sentinels was significantly strengthened in the kimchi groups. Which specific cells saw the biggest changes?
The effects were concentrated in 3 key APC types. Monocytes, which are these large white blood cells patrolling the blood and tissues.
Okay.
Dendritic cells, which are the most potent, the most professional antigen presenters, and even certain populations of innate-like B cells. The data showed these cells were genetically programmed to be better at their job. They were more alert.
So if kimchi gave the sentinels better binoculars, the next step is seeing how they communicated the threat. And this leads to the second core finding, a dramatic remodeling of intercellular communication.
This is where the computational analysis gets so cool. They used a tool called CellChat, which takes all that gene expression data from thousands of individual cells and reconstructs the conversational network.
So it maps who is talking to whom.
And how often, and the intensity of those signals.
So what did the placebo group's network look like after 12 weeks?
For them, the analysis showed that signaling strength in those key APCs, the dendritic cells and monocytes, it actually tended to decrease over the 12 weeks.
It went down.
Yeah, which might suggest a kind of natural slowing or slight fatigue of the immune surveillance system over time without any intervention.
But the kimchi groups flipped that trend completely.
Completely. In both kimchi groups, the immune communication network was selectively expanded and reinforced. We're talking a significant increase in both the number of interactions, more cells talking to each other, and the intensity of that signaling.
So the APCs weren't just better at their job, they were also communicating their findings more frequently and more loudly.
Yes, across the entire network. The whole surveillance system became much more interconnected and effective.
This tightened network is crucial because those signals are being sent to the cells that orchestrate the long-term defense. the T cells. And the study focused specifically on CD4 plus T cells, the helper T cells.
Right, and the changes here just perfectly illustrate this precision effect. The CD4 plus T cells showed an accelerated but also balanced differentiation.
What does that mean, accelerated differentiation?
It means they were pushed along their maturation pathway faster toward two distinct and what seem like contradictory functional types.
Let's break down those two paths. The first one was toward effector phenotypes.
These are your defense cells, the cells that actively go to the site of an infection and coordinate the release of chemicals needed to eliminate the threat, whether it's a virus or bacteria. This is the firepower.
Okay, so that's the go signal. What was the second path?
The second, equally important path was acceleration toward regulatory T cells, or TREGs.
And these are the peacekeepers.
Exactly. They are responsible for turning off the immune response once the threat is gone. They prevent the defense cells from overreacting or attacking the body's own tissues.
Which is the root of autoimmune diseases and chronic inflammation.
That's right.
So why is accelerating differentiation toward both defense and regulation at the same time the ultimate sign of a successful immunomodulator?
Because it means the system is prepared for a rapid, robust response, but that response is inherently self-limiting. It's controlled.
It has a built-in off switch.
A guaranteed timely off switch. If kimchi only boosted the effector cells, you'd risk persistent, uncontrolled inflammation. By simultaneously speeding up the maturation of the TREGs, kimchi ensures the strong fight is kept in check.
And the analysis confirmed this.
Yes, what's called pseudotime analysis, which maps the cell's maturation journey, showed that the kimchi groups had their cells moving towards the final functional stages for both roles much faster.
Now, this is where we check that blunt booster theory again. If kimchi was just stimulating everything, we'd expect hyperactivity everywhere, right? Did they check the major killer cells?
They absolutely did, and this really confirms the targeted nature of the effect. They checked CD8 plus T cells, the main killer cells that destroy infected cells, and natural killer cells.
Yeah.
The single-cell analysis showed that the composition and the functionality of these major cytotoxic players, they largely remained stable, or they showed only minimal, non-significant changes.
That is the ultimate proof. Kimchi is being highly selective.
It's optimizing the decision-making and command structure, the APCs and the helper T cells, without, activating the nuclear option.
It's enhancing preparedness and regulation, not just putting the entire war machine into motion.
It's focusing on intelligent mobilization and balanced control. It's
Yeah.
fundamentally different from a general inflammatory stimulus.
Okay, so we've established what changed at the cellular level. APCs got smarter and louder, helper T cells matured faster and in a more balanced way. Now we have to answer the deepest question.
Yeah.
How? How did kimchi pull this biological lever?
The mechanism it hinges on the dramatic upregulation of the major histocompatibility complex class II signaling pathway.
MHC class 2.
Yes, in those antigen-presenting cells, especially the dendritic cells, this is the epicenter of kimchi's effect.
Let's just refresh the role of MHC class 2. These are specialized proteins that sit on the surface of the APCs, right?
Correct. Imagine them as small, specialized display cases. When an APC processes an invader, a fragment of a virus, it loads that fragment onto the MHC class 2 molecule and physically displays it on its surface.
And this display is what the CD4 plus helper T cells then read to decide if they should activate the big immune response.
Exactly. The better the APC is at loading and displaying these fragments, the stronger, faster, and more specific the immune response will be.
So enhanced MHC class 2 signaling means better, more efficient intelligence sharing.
Precisely. The single-cell sequencing showed a robust enhancement of this pathway. They saw the upregulation of key genes like HLA-DRA and HLA-DRB1, which are the blueprints for making these MHC class 2 display cases.
So the cells in the human participants were literally making more of the machinery for antigen presentation after eating kimchi.
That's what the genetic data showed. But they knew they couldn't just rely on gene expression. They needed to prove that turning on the blueprint actually translated into better performance.
So they took it to the lab, the in vitro validation.
Exactly. They used laboratory models, THP1 macrophages, a type of immune cell, and exposed them to factors from the kimchi powders. They ran a functional test using a fluorescent antigen.
And what did that test show?
Well, this fluorescent antigen only lights up when it's successfully processed and broken down inside the cell, which is the step right before being loaded onto MHC class 2. And the kimchi-treated cells showed significantly enhanced antigen uptake and processing. On top of that, they measured the physical amount of the MHC class 2 molecule on the cell surface, and it was significantly higher.
So it confirmed that the genetic changes they saw in the people translated directly into better functional performance in the lab.
It was a perfect match.
Okay, we know the target is MHC class 2. Now for the scientific core of this deep dive, the actual switch. This brings us to the JAK-STAT1 C-IDA axis. This is the pathway kimchi is manipulating.
Right. This molecular pathway is the master regulator for inducing those MHC class 2 genes, and it's at the heart of our defense against things like viral infections. It's driven by a powerful signal called interferon gamma.
So interferon gamma activates the JAK-STAT1 axis, which then tells the cell to make more MHC the class 2.
Exactly. But here is the critical distinction that reveals kimchi's precision. When immune cells are exposed to a standard excessive inflammatory stimulus like bacterial toxins in the lab, it causes an excessive activation of this axis.
Overactivation.
Yes, hyperphosphorylation. It overactivates the JAK and STAT1 molecules, and that uncontrolled high-level signaling can lead to the very chronic inflammation we've been talking about.
It's like slamming the accelerator pedal through the floor. The system is turned on way too hard.
That's the perfect analogy. The kimchi supernants, however, they achieved a mechanism of attenuation. They didn't prevent the activation.
They moderated it.
And moderated it. When they treated cells with kimchi factors at the same time as the inflammatory stimulus, the kimchi actively lowered that hyperphosphorylation back down to an optimized, healthy level.
So it's not turning the system off, but it's preventing the overshoot. It brings the signal back into a Goldilocks zone.
Precisely. And this optimized, calibrated level of signaling was sufficient and necessary to strongly support the expression of the master switch gene, C-IDA. By regulating JAK-STAT1, kimchi effectively controls the master switch for antigen presentation, ensuring optimal defense without the inflammatory side effects. It's an elegant action.
How confident were they that this specific pathway was the main mechanism? They must have run an experiment to prove dependency.
They did. They used a specific pharmaceutical agent called ruxolitinib. It's a potent, clinically used inhibitor of JAK12.
It's a molecular off switch for this pathway.
It is.
And what happened when they used it?
When they pre-treated the cells with this drug, it completely abolished the kimchi-induced upregulation of the MHC class 2 genes.
A definitive proof.
Absolutely. Functionally, it also drastically reduced the enhanced antigen-presenting activity they had observed. So if the inhibitor turns off the kimchi effect, the pathway has to be necessary.
So JAK-STAT1 is the required molecular pathway for kimchi's major effects.
Correct. It's worth noting, though, that they did see some minimal residual activity, which suggests that the complexity of the whole kimchi matrix likely involves other auxiliary pathways. It's never just one switch, but this is definitely the main one.
Let's turn to the Trojan of optimization. The study compared the traditional, spontaneously fermented kimchi, the SK group, with the scientifically optimized starter culture 1, the LMSK. Did that high-tech starter culture actually prove to be superior?
This is one of the most fascinating nuances in the study, and it says a lot about the challenge of translating lab findings to the human body. In the controlled in vitro lab settings, the answer was a clear yes.
So in the Petri dish, the engineered kimchi, LMSK, was the better tuner.
Absolutely. It showed a more pronounced immunomodulatory effect. It was better at strengthening antigen recognition, and, crucially, it was significantly more effective than the spontaneously fermented SK at suppressing that excessive JAK-STAT1 phosphorylation.
So it was the superior precision device in the lab.
Yes, inducing higher levels of those vital MHC class 2 genes.
Okay, so we have a clear theoretical winner, but when they looked at the single-cell data from the human participants, did that superiority hold up?
Surprisingly, no. When looking at the changes in the cell-cell interaction networks and the key T-cell differentiation paths, the differences between the SK and the LMSK groups were broadly similar.
Wait, really?
Yes. Both were dramatically superior to the placebo, but the clinical distinction between the two fermentation methods was largely neutralized in the context of the human body.
So if the optimized probiotic strain was demonstrably better at hitting the target in a cell model, why would that advantage just disappear when a person eats it? That seems to undermine the whole push for engineered probiotics.
That's a necessary critical question. The data suggests that the incredibly complex human ecosystem, our own existing gut microbiota, our unique physiology, acts as a powerful buffering system.
A buffering system.
Once the kimchi is consumed, the overall matrix of the food, including the fiber and metabolites produced by our own resident gut bacteria, it seems to dominate the regulatory effect.
So even if the starter culture gives the initial product a molecular edge, the human gut biome, essentially equalizes the systemic benefit between the two high-quality kimchi types.
That's the interpretation. It strongly suggests that for these kinds of sophisticated effects, the benefits might derive more from the whole diet effect, the complexity of the fermented cabbage, salt, and spices, rather than the superiority of any single inoculated strain once it hits the human system.
It reinforces the power of traditional complex whole foods.
It really does.
The lead researcher, Dr. Wu-Jae Lee, he underscored this dual effect, activating defense and suppressing excess response, as the main breakthrough.
Right, and he emphasized that the World Institute of Kimchi is committed to expanding this international research on kimchi and its lactic acid bacteria in relation to both immune and metabolic health.
The real-world implications here are far-reaching. Confirming kimchi as a functional food with this level of molecular detail, it could really drive the development of functional health foods designed for immune balance.
Moving beyond the simple more bacteria is mindset you see with a lot of probiotics.
But the most intriguing potential application, stemming directly from this enhanced APC function, it relates to things like vaccination.
This is a critical point. Vaccines work by introducing an antigen to the immune system. That whole process relies on the efficiency of your antigen-presenting cells to ingest, process, and display that antigen to your T cells.
So if kimchi consumption optimizes APC function, as this study showed,
it could theoretically lead to a significantly more robust, faster, and longer-lasting immune memory after you get a vaccine.
a simple dietary intervention could enhance the effectiveness of a complex medical one.
Exactly. You're looking at a way to optimize the body's hardware so that the software, the vaccine, runs more effectively. The potential for improving vaccine efficacy is a massive and exciting avenue for future research.
And of course, the balancing act suggests applications in preventing immune-related diseases involving that chronic low-grade inflammation.
For sure.
Now, while this research is a landmark achievement, no study is perfect. For our listeners who want to be well-informed, what are the crucial limitations we need to keep in mind?
The most significant one is a trade-off for the advanced tech they used. The small sample size for the single-cell analysis.
Right. 90 people in the broader trial, but only 13 participants per group for this really deep, expensive analysis.
Yes.
13 people is an incredibly small number when you're talking about human genetics. I have to ask, how can we trust that the dramatic changes they saw aren't just idiosyncratic responses of a few people?
That is a completely fair challenge. The scientific justification really relies on two factors. First, the design was a paired within-subject comparison. They compared the genetic profile of a person at week 0 to that same person at week 12.
So each person was their own control.
Right, which significantly increases the statistical power to detect changes caused by the intervention. Second, the mechanism they identified, that JXSTAT1 axis, was validated externally with the robust in vitro models and the inhibitor drug.
So the small size is robust enough to establish the mechanism, the how, but maybe not robust enough to establish the generalizability, the who.
Precisely. We have a solid mechanism of action, but we can't extrapolate these effects to the entire global population of all ages and health states just yet. It's a phenomenal proof of concept that needs to be expanded.
The second limitation is tied to the technology itself. The conclusions are based on gene expression data. We know what the cells were preparing to do. But did this study fully confirm the function of these remodeled cells in the living human body?
That's the gap. The study lacked comprehensive functional validation in the humans themselves. They didn't include measures like flow cytometry on the blood samples, which would physically confirm the surface expression of MHC class II and the actual proportions of those newly matured T cells.
Those are very detailed blueprints, but we still need the final photograph of the finished house built in the human body.
That's a great way to put it. And finally, there is the cohort homogeneity. The participants were a very specific group, overweight adults whose immune systems were already slightly altered by metabolic factors.
Which means we can't assume that a very lean, healthy 20-year-old would see the exact same magnitude or type of immunological balancing, right?
That's right. For this group, the regulator mechanism might have been focused on reducing baseline inflammation while enhancing defense. In a healthy, non-inflamed person, the effect might be solely centered on optimizing antigen presentation.
So its application to other populations is a critical area for follow-up research.
It is. This study identifies the switch, but future studies need to see how strongly to flip that switch in different types of people.
This deep dive really forces a shift in perspective. We've moved way past the idea of kimchi being a simple gut health aid. This research has confirmed it's a profoundly sophisticated dietary immunomodulator.
Mechanistically, we've learned it doesn't just stimulate, it fine-tunes. It uses its complex metrics of ingredients to target the JAK-STAT1 signaling axis, precisely enhancing antigen presentation and ensuring the helper T-cell response is both robust for defense and tightly regulated. It promotes intelligent mobilization.
The convergence of ancient dietary wisdom, rooted in something as simple as preserving cabbage, and our ability to analyze its effects with single-cell gene sequencing is just a stunning achievement.
It really underscores the immense hidden complexity of whole diet interventions when compared to the more reductionist approach of using single molecules or single-strain supplements.
And if a centuries-old dish harbors such a complex, precision-tuned regulatory mechanism for our most vital defense system, it prompts a really important question.
What other traditional, culturally complex fermented foods, from kefir to sauerkraut, natto to injera, might also harbor similarly potent, high-resolution effects, just waiting for scientists to apply the right tools to uncover their secrets?
This study is a powerful reminder that our diet communicates with our immune system at the deepest, most molecular level. We'd encourage you to continue exploring this burgeoning field of nutritional immunology and to recognize the powerful, precise signaling contained within the foods you eat every day.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.